Hello, it's my pleasure to welcome you to this virtual scientific meeting developed by the American Neurogastroenterology and Motility Society, entitled What's Behind the Positive Effects of Neuromodulation on Gut Function. My name is Beverly Greenwood van Meerveld, and I'm the immediate past president of the ANMS and current director of the ANMS Institute. And before we start tonight's event, I'd like to remind attendees of a few important dates. I'd like you to save the date of August the 11th to 13th for the ANMS annual meeting in Austin, Texas. We will have the clinical course as well as the scientific meeting. An important date to remember is the abstract deadline is March the 8th, 2023. And this meeting will be preceded by the ANMS Young Investigator Forum and the ANMS Fellows Advancing Motility meeting. And this is specifically for second and third year fellows. And these meetings will be on August the 9th and 10th. But for more information, please visit the ANMS website. I'd also like to mention about the ANMS Basic Science Mini Sabbatical Program. This is where applicants travel to a sponsoring lab at an outside institution to learn a new research method or model system. They're 2 to 4 weeks in length, and you get support, $2,000 each for the applicant and the sponsor. So please consider submitting an application for support in 2023. So it's now my great pleasure to introduce today's moderators. First, Dr. Ken Koch, who is Professor of Medicine in the section of Gastroenterology and Hepatology at Wake Forest University School of Medicine and Atrium Health. He has a long interest in the electrical activity of the gastrointestinal tract, especially the pacemaker system that controls the stomach and neuromuscular function. He has published over 175 scientific papers, 3 books, and contributed over 40 book chapters. His dedication to research continues as he investigates the causes of nausea and vomiting, and he's also studying specific diet, drug, and device treatments for patients suffering from neuromuscular diseases and disorders of the GI tract. I'm also delighted to introduce our second moderator, Dr. Jen Wellington. Dr. Wellington is Assistant Professor of Medicine at Wake Forest University School of Medicine and an Assistant Program Director for the Atrium Health GI Fellowship. She completed a T32 GI Fellowship at the University of Maryland School of Medicine, where she earned a master's degree in clinical research and epidemiology. Her research interests are in gastroparesis, pyloric neuromuscular dysfunction, and fecal incontinence. She joined the faculty at Atrium Health in Charlotte, North Carolina in 2021, where she practices as a neurogastroenterology and motility specialist for comprehensive motility disorders. Additionally, she has an interest in graduate medical education and curriculum development for trainees at her institution. I'd now like to turn over tonight's event to Dr. Koch to introduce our first speaker. Thank you, Ken.
All right, Beverly, thank you for the introduction, and it's my pleasure to introduce Dr. Chen in a moment. I do want to ask our participants to send any questions that come up during these talks to the Q&A portal. Just send them in there, and we will address them in the Q&A section that's coming up. So keep those questions coming in, please. Well, Jiande Chen is Professor of Medicine and Director of Neuromodulation in the Division of Gastroenterology and Hepatology, and also has an appointment in the Department of Bioengineering at the University of Michigan. He has had over 110 research grants from various organizations through his career. He has trained more than 100 doctoral students, postdocs, young scholars, has more than 30 patents, and published more than 450 peer-reviewed papers. He was the first president of the International Electrogastrography Society, and he's presently the vice president of the International Gastrointestinal Electrophysiology Society. He has held council member positions at our own ANMS and has served on many editorial boards of peer-reviewed professional journals. Dr. Chen's current research interest is in the area of neuromodulation and applications to treating obesity, diabetes, the functional gastrointestinal diseases, and inflammatory bowel disease, for some examples. So we're very excited to hear his comments today. The title of his talk is Transcutaneous Auricular Vagal Nerve Stimulation Improves Functional Dyspepsia by Enhancing Vagal Efferent Nerve Activity. So Jiande, we look forward to your lecture.
Thank you very much, Ken, for kind introduction. And also thank the organizers for inviting me to give a talk about neuromodulation. Now let me share the screen. So first, let me go over the method we can use for neuromodulation. So first, we can directly electrically stimulate the gut, like the stomach, intestine, and colon. So Dr. Abell is going to talk about the gastroelectrical stimulation. And secondly, we can directly stimulate the nerve, like vagus nerve and sacral nerve. These are autonomic nerves, and can also stimulate the spinal cord, that's the sympathetic nerve. And today I will be talking about non-invasive neuromodulation method, like by stimulating the surface of the body, such as we can stimulate the acupuncture point, or we can stimulate the auricular vagus nerve, or you can stimulate the transcutaneous, the cervical vagus nerve, or tibial nerve. So the common ground for gastrointestinal neuromodulation is that we know that we have the central nervous system, we call that the bigger brain, and then we have anterior nervous system, we call that the little brain. So between the big and the little brains, there's autonomic nerves. So it is divided into parasympathetic nerve and sympathetic nerve. So we all know that in patients with functional GI disorders, they have reduced vagal or parasympathetic activity. And a lot of times they have overactive sympathetic activity. So what we can do with neuromodulation is that we can do one or both of them. One is to enhance parasympathetic activity. The other is to inhibit sympathetic activity, or you can do both. So that's a common ground. So transcutaneously, we can stimulate like acupuncture points, or we can stimulate the nerves which are superficial. So recently, we have been working a lot on a method I call that the transcutaneous electrical acute stimulation. So this method has several unique features. One is that the stimulation is applied via acupoints that are in the vicinity of peripheral nerve, such as auricular acupoints, the topic of today. And we can also stimulate a point on the leg called ST36, which is in the vicinity of several nerves like a perineal nerve, sciatic nerve, and tibial nerve. Or we can stimulate a point like a relief band, the wrist point here. It's called a PC6. So this is coincide with median. So that's one feature. The second feature is that we have to use specialized or optimize the parameters. It's not like you just apply electric coolant and you think you can hit the target and solve the problem. So in our lab over the years, we have derived a few sets of different parameters which are good for treating different symptoms or different pathophysiologies, such as 25 Hertz for stimulating or improving gastrointestinal motility, and 100 Hertz for improving visceral pain, or 5 Hertz for improving inflammation. The third feature is that we will apply electrostimulation daily, not like acupuncture. You do it only a few times 1 week. But here, with that, with non-needle or needleless stimulation, patient can do that at home. So we can do daily administration, and this way we can enhance the efficacy. And lastly, we have to use a wearable device. It's either wired, like here, or we can use a wireless device. So these are something we are actually developing, like using this device. You can just patch it, or for auricular stimulation, you just plug it like put it into the ear like a Bluetooth. And for risk stimulation, you can just wear it like a watch. So these are topics we have been collaborating with various companies and try to develop a non-invasive device so that it can be used more easily. It can be used more conveniently by our patients. So today I will be focused talking on one particular method, that's transcutaneous auricular vagus nerve stimulation for functional dyspepsia. So for this audience, probably we all know that the pathophysiology of functional dyspepsia. Although it's not complete, but we know there's a problem with impaired gastric accommodation. Proximal stomach does not relax, and the visceral organ, the stomach, is too sensitive. We call it hypersensitive to distension, so that the patient have pain. And also there's a problem with the pacemaking activity, like a gastric dysrhythmia and a delayed emptying. Then in the duodenum, there's several things like duodenal mucosal alterations, like increased permeability and so forth. So today we will show whether TAVNS can improve some of these abnormalities. So this was a study conducted a few years ago, actually in China, by one of my doctoral students. So we involved 33 patients with functional dyspepsia and then divide them into two arms, one treatment with TAVNS. So we apply bilateral electrostimulation via thymboconcha, and then another group we stimulated it via the arm, okay, for 2 weeks. Then every day we deliver 2 times of stimulation lasting for 1 hour. Then at the before and end, then we will assess several things I will discuss further. So the stimulation parameter is very important, but I will not discuss here. But these parameters were derived from various previous animal studies and preclinical studies. So as I said, we delivered electrostimulation via this point or via this point twice daily, each lasting 1 hour using this set of parameters. So the amplitude is adjusted so that the patient can feel it, but they can tolerate it. And then at the beginning and end of the study, we performed, I call that a 3-in-1 test. So we will record EGG using a 4-channel device. At the same time, we record the EKG signal for 30 minutes. And then we ask a patient to drink nutrient liquid until completely full. So that's a satiety test. And then afterward, we will continue the recording of the EGG and the EKG for 30 minutes. And during that 30-minute period, we will assess patient's symptom on fullness, nausea, and pain at a 10-minute interval. So let's show the result. First, we look at the dyspepsia symptom score, and we can see that in the sham group, we don't see a significant difference, but in the treatment group, we see a significant decrease. And we look at the fullness right after a satiety test. So you can see that during no difference at the beginning, but at 20 minutes after the drink, with sham stimulation, these two groups, you don't see a difference, but with stimulation group, you can see a substantial reduction. And at the 30 minutes also. Bloating score was reduced significantly with TAVNS, but not significantly with sham stimulation. And the pain, although the pain was mild, but the reduction was significant. And interestingly, the score for depression and anxiety was also significantly reduced in the treatment group. Now let's look at the pathophysiology of functional dyspepsia. So first we look at the maximum tolerant volume. So that's the drink test, right? So in normal subject, we actually in that study, we also did a normal subject. we studied 39 healthy volunteers. So normal subject can tolerate about 1 liter or 1,000 volume. But in patients, they can tolerate about less than 800. But after chronic stimulation, that value was increased to 900. And there was a minor or more very mild but a significant correlation between the fullness score and the maximum tolerant volume. And then we look at the gastric slow waves. You see that the slow wave at the beginning of the study, it was about 60%. So that was actually lower than in healthy volunteers. In healthy volunteers, it was about 80. But after 2 weeks of treatment, we can see that in both fasting state and fed state, there was a significant increase with the treatment. So if you would like to know how we analyze the slow waves from the electrogastrogram, this is the method. We got the recording, and then every minute we calculated the dominant frequency of each minute. And then we will count how many peaks are within 2 to 4 cycles per minute range. Then we got the percentage of normal slow waves. And we also find that the normal slow waves, that means the higher percentage of normal slow wave is mildly associated with higher volumes the patient can ingest. And also negatively correlated with the fullness score. So that means that if the gastric pacemaking activity is more normal, then they would have lower score in fullness, and they could tolerate a higher volume of ingestion here. So now let's look at the mechanistic things about because we are stimulating the auricular vagus nerve, right? So we are hoping that the stimulation itself can enhance the vagal efferent activity via the vagal efferent pathway to the brain. So we assess the non-invasively from the EKG signal. This is the method we use. So we have EKG signal recorded, and then we detect each RR wave. Then from the RR wave, we have the beta to beta interval, right? So we convert that into a heart rate signal. Then from the heart rate signal, we perform a spectral analysis. Then we have low frequency component, then we have high frequency component. So high frequency component has been validated to reflect purely vagal efferent activity. Now let's look at the result here. So you can see that in the fasting state with sham stimulation, these 2 bars, there was no change. But with vagus nerve, transcutaneous auricular vagus nerve stimulation, you see a significant increase in high frequency component. That means increase in vagal activity, and in the fed state, that's the same. And we also found a mild but significant correlation between vagal activity and the percentage of normal slow waves, meaning that if the vagal activity was higher, then the slow wave was more normal. And if we look at the ratio between sympathetic and parasympathetic activity, so that represent a higher value, means that a more sympathetic dominance. So you can see that no change with sham stimulation, but with real auricular vagus nerve stimulation, we see a decrease in sympathetic dominance in the fasting state and also in the postprandial state. And that ratio was negatively correlated with percentage of normal slow waves. So these data suggesting that enhancing vagal activity may improving the pacemaking activity and may eventually improving motility. Unfortunately, in that study, we didn't perform motility. We all know that the actual assessment of antral contraction is too invasive, and most of our patients cannot tolerate it. So that's why we use the EGG, electrogastrogram, as a surrogate of antral motility. So in their study, only 1 out of 36 patients reported mild tinnitus and insomnia, but these patients, their patients still continued to complete the study, and there was no sequelae. No other side effects were reported. And all 36 patients completed the clinical trial. So it showed a very good safety profile. So now let's discuss about the possible pathways and mechanisms. So most of these were derived from our basic research, because in human clinical studies, we are very limited, okay? So what we found from basic animal research was that when you stimulate the simple contour, actually you can activate the nucleus tractus solitarius in the hindbrain. Okay, and in animals, we can look at the C-force expression during acute and chronic stimulation here, and we found a substantial increase in C-force expression in NTS. And then we know NTS is connected with the dorsal motor nucleus of the vagus nerve, and then it will generate an enhanced vagal activity. So that's the pathway we hypothesized. So from auricular contour to the nucleus tractus solitarius, and either directly projected to DMV or through other part of the brain, and then come to the DMV and generate an enhanced vagal efferent activity, and then release the acetylcholine. And released acetylcholine will lead to improvement in motility. So that's the mechanism for improved slow wave activity in this study. But in animal studies, actually, we could assess, gastric motility. We could also assess gastric emptying, and we found that this kind of method actually does improve motility in addition to slow wave. But we also showed that the maximum tolerable volume was significantly increased, right? So that's a suggestion that the stomach can accommodate a higher volume of food. So that effect, we believe that it was mediated via the release of nitric oxide. So we also had animal studies, although we don't have that in this current clinical study. And also we showed that there was an improvement in abdominal pain, although in that group of patients, the pain score was not very high, but the improvement was quite significant. So we hypothesized that the improvement in abdominal pain probably was attributed to the reduced inflammation via the cholinergic anti-inflammatory pathway. We all know that, release of acetylcholine would result in the reduction of inflammation cytokines via the alpha-7 nicotinic cholinergic receptor. And we also have animal data to show that, TAVNS or auricular vagus nerve stimulation can improve epithelial barrier function, and it can also reverse the efferent sensitization like DRG neurons. So these are our basic animal studies. And we also show that in this study, the anxiety and depression score was also significantly reduced. And we believe that this could be attributed to the central effect of TAVNS, probably by altering certain brain functions like ACC or amygdala, and also probably by improving functional connectivity. So these are the topics we are currently working on in our lab. So in addition to auricular vagus nerve stimulation, we also performed quite a few dozens of studies using electrostimulation via the acupuncture point, like the S236. And there have been a lot of publications in the literature from our group and also from other groups for treating various functional GI disorders like GERD and gastroparesis, irritable bowel syndrome, and constipation. And for surgical recovery, because post-surgically, there was an increase in sympathetic activity, and there was a reduction in vagal tone, and there's dysmotility, right? So there are several publications showing the improvement of post-surgical recovery in patients after various surgical procedures. And there were also publications showing that this kind of stimulation, non-invasive neuromodulation, may also reduce inflammation and acute pancreatitis and radiation proctitis. And also for pain, although in the current study I presented, the pain was mild, but there was a lot of, there were ample data in animal research, and we showed that there was an antiemetic effect via using TAVNS or TA. That means you place the electrode at the body point of acupuncture. So in conclusion, transcutaneous auricular vagal stimulation is effective, although this is a single center study, in treating functional dyspepsia by improving passive physiologies of functional dyspepsia via the vagal-vagal pathway. One interesting feature of TAVNS is that it has integrative effects on various passive physiology factors of FD, such as dysmotility and pain. We know that clinically it's very difficult to treat patients with both pain and dysmotility, because when you're treating pain using certain pain medications, and most of them will worsen motility, right? Or if you want to improve the relaxation of the stomach, and then you would worsening dysmotility in the antrum. But in this study, we found that it can improve accommodation, but it can improve the gastric slow waves, and at the same time improve pain. So that is a very interesting feature. But I should emphasize that stimulation modality is critically important in clinical application, including stimulation locations, parameters, and treatment regimen. So these cannot be coming, from the air. You have to figure out very carefully and using the appropriate regimen. For patients with severe symptoms, it may be used in combination with our conventional medical therapies. However, to prove its clinical efficacy, probably we need a multi-center study to further validate this single study. And also more research is needed to investigate its mechanism of action. So finally, I would like to thank the team who performed that study. So the study was published in American Journal of Gastrointestinal Physiology. And for the TA study, transcutaneous electroacute stimulation study being performed clinically and also in basic lab, I would like to thank support from NIH. And finally, but most importantly, I would like to thank my team who conducted all these clinical and basic research studies. Thank you very much.
Thank you so much, Dr. Chen, for your presentation. That was so interesting. Thank you very much. First, I'd like to thank the ANMS for this opportunity. It's my distinct honor to introduce a longtime role model, Dr. Abell. Dr. Abell is the Arthur M. Schoen, MD Chair in Gastroenterology at the University of Louisville, where he's been since 2012, continuing his more than 30-year instrumental career in setting up GI motility clinics in the Mid-South. Dr. Abell has had a prolific career in research in all types of motility disorders, with a special emphasis on gastroparesis syndromes, and is an original member of the NIH Gastroparesis Clinical Research Consortium. Dr. Abell has a long interest in GI manifestations of neurological disease, including spinal cord injury, for which he is involved in several research studies with the NIH. His team in Memphis pioneered the current technique that we use for GI neuromodulation of the upper GI tract, and the use of temporary gastric stimulation has been a focus of his work in Louisville, which actually currently implants about 10% of the world's permanent GI bioelectric devices. That's an incredible feat. Dr. Abell's newest NIH work involves high-resolution gastric electrical body surface mapping as a diagnostic tool for GI motility disorders. Please join me in welcoming Dr. Abell, and we are all very excited to hear about your work today.
Well, thank you for a kind introduction. I'll try to get this done succinctly so we have a little time for discussion. Let me start by sharing the screen, and we'll get started. Thank you again for this opportunity to talk today with this distinguished group. I'm going to talk about electrical stimulation of the stomach on gastric mapping and small bowel motility in patients with gastroparesis and concomitant pancreatic and small bowel dysfunction that I'll talk more about later. Here's my disclosures, NIH and other work. But most of what we're going to talk about today was not externally funded. I'm going to talk about the background of electrical stimulation briefly for a couple minutes, including some work on the pancreas, and then on neuromodulation and methods in animals and humans, then the results in animals and humans, and then a couple minutes on discussion. So bioelectric neuromodulation includes many things, as Dr. Chen mentioned, but gastric electrical stimulation, or GES, is one of those. The mechanisms are increasingly well understood and beyond the topic of this presentation, but I'd like to talk a little bit about GES effects on the pancreas, and then look about how we can evaluate small bowel slow waves using different energy dosing regimens. Electrical stimulation goes back over 100 years to about 1908 or so. The recent era included work by Keith Kelly, who was one of my mentors, and colleagues at Mayo Rochester. Slow wave dysrhythmias are associated with gastroparesis in multiple models, as it was alluded to earlier, and GES with both low and high energy and many energies in between have been used. This is an algorithm of current recent publication of what we do clinically with someone with the symptoms of gastroparesis, ruling out mechanical obstruction, defining physiology, looking at nutritional and drug therapies, but then we get down to the drug refractory patients, which may get pyloric therapies, may get enteral tubes, may get other testing or assessments. But I'm going to talk today about the bioelectric therapy of electrical stimulation of the stomach. This is an article in Press in Neuromodulation that's looked at almost 800 patients in the 9 control studies of stimulation. And this is looking at various lengths of the studies, mean differences between patients and controls. And you'll see if all the studies, actually except one, were positive in the effect. And this is the first look at a really large number of patients. I'd like to just go back for a minute. 25 years ago, when electrical stimulation was still investigational, and this part was supported by Medtronic, we were looking at electrical stimulation and effects on the pancreas, looking at pancreatic elastase, which was relatively new then, fecal elastase. And we did a study with randomized control study with device on and off, and we also looked at some control groups, also measured polypeptide levels, GI symptoms, and heart rate variability. And the reason I mentioned this is because this was kind of the background of how we got into today's work. But here we knew that pancreatic elastase was low in people with gastroparesis, whether they were non-responders or responders. But we showed that elastase goes up with stimulation, permanent stimulation, and pancreatic polypeptide, which is a vagal marker, goes up a little borderline. P-value 0.08, but it's clinically, I think, significant. And one last slide about this. If you look at 24-hour Holter heart rate variability, maximum minimum heart rate, the stimulator on the heart rate goes down, and the symptom scores go down almost identically. So it suggests that part of the effect here is vagally mediated by the pancreas. So we know about the pancreas. The question is, could we change small bowel electrical activity? And so we did a pilot animal model and a very pilot human model. And I'd like to talk about that in just a moment. The gold standard for small bowel motility is still the gastroduodenal manometry, although one could argue that the full thickness biopsy might be the true gold standard. Gastric electrograms can be adapted to the small bowel, which we've done, and we tend to look at frequency, amplitude, and propagation, whether it's low resolution or high resolution. And we're going to use both of those in the animal model and the low resolution human model. One more slide on the electrogram or EGG, if it's cutaneously. The single points in the 70s led to low resolution in the 80s and 90s, and then high resolution now cutaneously. We're going to be using low resolution mucosal and high resolution serosal and low resolution mucosal as well in the animal and human study today. So again, this is our study. I'd like to acknowledge my colleagues, really national and international, because the human work was done here in Kentucky, but the animal work was done in Mississippi, and some of the high-res work was done with colleagues from New Zealand. So first, the methods on animals. We had an aim of looking at the relationship between the gastric electrical stimulation delivery and small bowel and biliary slow wave activity, and used for this a evoked response in a non-survivable swine model, a very small sample size. In this pilot, we used low and high resolution electrical slow wave mapping for frequency, amplitude, and the ratio of both duodenum and sphincter vodi. And a couple of illustrations how we did that. This illustration on the left is electrical device put in the pig stomach, and then electrical recordings, both high and low resolution in duodenum and sphincter vodi. This is a picture on the right of a laparotomy, low resolution electrodes. And the high resolution grids that we use can contain up to 256 electrodes that sit directly on the serosum, and we're able to do the recordings simultaneously. Again, in the animal model, we looked at small bowel and biliary recordings for frequency and amplitude and their ratio, visually analyzed by 2 investigators, and we used the additional high resolution grids for small bowel simultaneously. We used a linear mixed model for multiple comparisons. Also, in the animals, we use what we've done and published in humans, 3 different energies in rotation for 5 to 10 minutes each, low, medium, high. The differences being current, 5, 10, or 20 milliamps, rate 14, not stated there, but 14, 28, or 55 hertz. And the pulse widths vary between 330 and 450 microseconds. And then it was a tenth of a second versus 1 second, 4 seconds on. So there are marked differences in the energy that that's developed here if you measure it, for example, in joules. So what about the human pilot? Well, the aim of that was to look for improvement in symptoms associated with proximal small bowel dysmotility with gastric stem. And we had 8 patients from a large cohort that's prospectively obtained who underwent temporary electrical stimulation with gastric and small bowel electrodes. We used subjective and objective data before and after temporary stimulation for approximately a week. We looked at symptom scores, gastric emptying times, solid and liquid, mucosal electrograms using low resolution mapping, and also a high resolution health-related quality of life, as well as our low resolution mapping. A couple more things on the methods for humans. 2 leads in the stomach, 1 proximal recording and 1 distal for stem. The distal stem electrode used as an augment of the nominal energy, so it's basically nominal energy. Recording electrode was in small bowel right near sphincter vodi, which is, as you know, is pretty easy to reach endoscopically, and we recorded mucosal EGGs, low resolution EGGs from the proximal stomach, the recording lead, and the small bowel sphincter vodi. And those again were measured for 5 to 10 minutes at baseline after 6 days of recordings for frequency, amplitude, and ratio. And these were done with 2-sided paired t-tests. Again, a little bit more on the adult data. The patients had to have at least 1 of these or more, dilated small bowel, idiopathic diarrhea, or idiopathic pancreatitis. And we were very interested in the type of pancreatitis, which I won't detail today, but that's another focus of ours. We did, again, our 6-day recordings, FDA compliant scores, solid liquid emptying, mucosal EGGs, and a Medicare-derived health-related quality of life measure. These are the 12 patients. Again, there was only 1 with dilated bowel, 2 with small bowel diarrhea, 5 with pancreatitis. Here's the type of pancreatitis. 3 had reflux symptoms, and we also quantified some other things related to pancreatitis. And now going on to the results, and I think we're doing okay on time here on results. Here's on our animal models, duodenal frequency, amplitude, ratio, sphincter Voti frequency, amplitude, and ratio. These are the p-values with a mixed linear model. You'll see none of these was significant, although amplitude approached borderline significance. One will see is that in general, frequency changes in the small bowel and in sphincter with the device being activated, and the amplitude also changes, and that's probably better on this next slide. Looking at frequency, here's duodenal frequency again, baseline in blue, low, medium, high energy, and some changes in frequency and amplitude. We wanted to see was whether we could change these, and whether there might be intrinsic neural connections. This wasn't significant, but you'll see in high energy amplitudes higher, particularly in sphincter Voti. So with this baseline animal data, we also collected high resolution recordings. This was fairly early days with high resolution. And here's baseline, low, medium, and high. And there are differences here, although we did not quantify these as we do now for significance. And there are also differences in waveforms between duodenum and sphincter Voti, which we published separately in humans. Sphincter Voti, pancreatic sphincter, and biliary sphincters all have distinct electrical patterns, which you can detect, and I used to determine back when I was doing ERCPs. So we were able to do this simultaneously with the low resolution recordings and the porcine model, pig model. So let's go on to the results for a human study, looking at slow waves, which is what we wanted to do in gastric emptying. The proximal gastric frequency and amplitudes are here. The small bowel frequency and amplitudes are here. And what we see is what we see routinely. And we published almost 1,000 patients last year in neuromodulation with this technique, 991 patients. Small bowel frequency and gastric frequency goes down significant here. Amplitude was not significant, but it's certainly a trend to go up in both. Gastric emptying, interestingly, was not significantly different. But there are some things worth commenting on. The 4-hour solid emptying improves as it usually does. But interestingly, in these patients who did not have delayed liquid emptying, did have delayed solid emptying, there's a slowing in the first 3 hours. And we found that correlated with the patient's small bowel diarrhea, those that had diarrhea, improving. What about symptoms in the human study? Well, here's their patient-reported outcomes in blue, baseline, green, and follow-up. Nausea, vomiting, pain, and total symptom score all improved, and diarrhea also improved. And there were some other changes that were not significantly different. So human results in summary, and then I'll go back to compare both human and animal in a few minutes for discussion. The patient-reported outcome symptoms, nausea, vomiting, pain, total symptom score, and diarrhea improved. And the mucosal EEG low resolution measure of small bowel frequency improved statistically significantly, and there was a trend in increased amplitude. The health-related quality of life score changed, which did not show you that, but it changed significantly, and all the changes were significant except gastric emptying. So what about the conclusions? Conclusions for the last couple of minutes. Animal and human studies. So in an animal model with gastric electrical stimulation, there's a non-significant effect in small bowel, slow wave activity, and myoelectric signaling, suggesting to us at least the existence of intrinsic neural connections. I'll talk more about that in a minute because others have shown this with mapping to be true. In the human data, there is more significance with possible potential for therapeutic use of electrical stimulation in the stomach in patients who have gastroparesis symptoms and pancreatic, biliary, and small bowel symptoms as well from mid-gut. The limitations are probably obvious. It was a non-survivable pig model, small sample size, in part because of our lack of external funding. Small sample size in humans, relatively small number of people that we see for our overall patient population. And the human study was also open label. So a couple slides on discussion before closing. GI bioelectric stimulation is used increasingly. Most people know about it, thalamoenclave, Parkinson's, and movement disorders, but it's most commonly done in the world of gastric and sacral stimulation, but also spinal stimulation. Intestinal stimulation that Dr. Chen has written about extensively shows promise, but human studies are limited so far. The GES effects are not limited to the stomach. They include autonomic and pancreatic function, which I did show briefly. I did not show effects on inflammation and hormonal changes, which we have published in Neurogastroenterology and Motility in the last few years. From the animal pilot, GES can induce measurable slow wave changes using both low and high resolution electrograms. And for the human pilot, gastric stimulation can induce symptom change, upper and mid hindgut with changes in small bowel frequency and amplitude. So again, a couple of discussion again, the animal pilot was too small to be definitive, but it would be nice if it could be replicated and see if it get the same results. High energy had the most effect. The human study was more conclusive in effects. The human energy used was just augmented over nominal settings, augmented slightly. Again, we think this suggests intrinsic neural connections, but needs to be validated in others. Within the GI tract, therapeutic potential for patients with small bowel or pancreatic biliary disorders exists. I'd like to thank my colleagues, my collaborators from around the world, staff, funding agency, especially patients. And I've included some references. I've alluded to some other references, and if anyone wants copies of those, in the discussion, we can talk about them, or I can be reached by email easily and be happy to send any references that I've mentioned. So let me stop that. Hopefully I've left a little time for discussion.
All right. Well, thank you, both of you, for those very novel and creative studies on neuromodulation of places we don't usually think of. And so Jen, as my co-moderator, do you want to start the questions yourself, or we have a number of questions that have been sent in as well. So Jen, I'll let you start if you like with the first question.
Absolutely. And just again, just a reminder to our participants, of which we have so many tonight. This is really a great turnout. We're starting our question and answer session, so don't forget to put your questions in the Q&A box, and we'll try to get to as many as possible. All right, so the first question I thought I would ask, it's practical, and it comes from Paul Yeaton. He put two questions in. So he asks, is it simpler to just have acupuncture? This is to Dr. Chen. And can the opposite effect be created to mitigate fundic distension to achieve weight loss?
That's a very interesting question. So actually, non-invasive neuromodulation is simple because patient can do it at home. With acupuncture, patient had to come to heart clinic or to an acupuncturist daily. If they don't come daily, they do it only once or twice a week. The result is very limited. So the second question about the gastric distension, yeah, it's very interesting. For patients with functional dyspepsia and gastroparesis, they have reduced accommodation. So we want to increase that to a normal level, like I showed, right? Normal volunteers, they can tolerate 1 liter, but a patient can tolerate only 600. After stimulation, we can increase them to 600, 800, or 900. So that's one area. The other area is that for treating obesity, actually, you can do it. Then you have to increase the gastric fundic volume from normal to super large. Then you can use it for treating obesity. Actually, we did a study before. Then the parameter will be totally different.
All right. Thank you. I've got a couple of questions here as well, perhaps one for each of you from our colleague Henry Parkman. And I think, Tom, maybe you take this one, but Dr. Chen talked kind of interesting about these 3 different stimulation parameters, 25 hertz, 100 hertz, 5 hertz, that might attack different problems from pain, motility, or inflammation. Tom, have you used any of those sorts of differences, or do you agree with those differences in what stimulation can do? And have you used those kinds of changes in your work with the Entera programming?
Short answer is yeah, we use a lot of different stimulation parameters for
I'm sorry, just a little bit louder, please.
stimulation parameters for Entera, including Jiande and I actually have a project together looking at some of those different parameters. So yeah, we use a lot of we use particularly a medium, what I call augmented medium energy, and some high energy. There are probably about, with any device, there are probably about 10,000 ways. Now that's not an exaggeration that you can program them. So there's a whole lifetime of work that needs to be done about what's the optimal stimulation parameters.
Yeah, so I mean, do you, and Jiande, your three different types of stimulation, is those based on your animal work, or is that also true in human? Does it translate?
Yeah, that's a very good question. So when you apply neuromodulation, you have to look at your effect. If you are looking at the efferent effect, that means that you send a signal to brain, that the brain send a signal, write the signal out to control the disease, right? Then the parameter is very important, because you have to send the right signal to the brain, so brain knows that, oh, this is a problem for inflammation. This may be a problem for pain. So that's why, if it's indirect neuromodulation method, the parameter is extremely important. So the parameter we have, like a 5 Hertz, 100 Hertz, 25 Hertz, these are derived based on animal studies. So typically we will spend half a year to find out the parameters. And most of them can be translated into in humans.
Yeah, that's super important. And it did one brief little question about that, because in most of the diagrams of the classic parasympathetic, sympathetic, there's not a big arrow to the ear. All the other there, the ear is not the most what we'd think of for the vagus nerve. It's the wanderer, but that's a little crazy. Do you think those are mostly afferent? I mean, do we know why the vagus finds itself in the ear, and is it mostly afferent? Do we know about those actual circuits into the brain?
Yeah, so the vagal nerve in the ear is 100% efferent, 100%. And then the innervation density is different in different areas. So the symbol contour is 100% innervated. If you go to earlobe, probably there's very little innervation there. So everything you do here, if you are really doing nerve stimulation, it goes to the brain. It's via vagal efferent. There's no direction projecting to the J tract. But there's some connection with the facial nerves, but not with J tract.
Yeah, well, that's where the connection, like you said, is the NTS, maybe other circuits that then somehow find their way through whatever the stimuli are to affect the stomach. Thank you.
That's right.
Jen, do you have the next question?
I have a question just following up on that. There are so many studies that have shown the use of TENS units, which I think are widely available for chronic abdominal wall pain, abdominal pain, patients with our pain predominant gastroparesis, which is a large population of patients that we try very hard to avoid opiates in. And so I wanted to ask you, Dr. Chen or Dr. Abell, either of you, if you found that there was a specific frequency that really targeted the pain symptom. I know that in your study, your patients mostly reported mild pain, but we do obviously have a spectrum of that pain scale reported in our gastroparesis and functional dyspepsia overlap.
Yeah, so there, superficially, you think the method I introduced, I call that transcutaneous electroactive stimulation, is very similar to TENS. But actually, conceptually, it's very different. With TENS stimulation, you apply stimulation at a location where you have pain. Okay, so with TA, with T or with TA VNS, you stimulate there, but you want to activate autonomic nerve, and your target is a remote area. So that's different. So the parameter is very different. And also the action, the pathway is very different. With TENS, but most of the time, probably it's more local effect, like a massaging of the muscle and also increase in blood flow and things like this. But with the method I introduced today, it's more in enhancement on vagal activity or parasympathetic activity. So that's a big difference. So in this current study, the pain was quite mild, but we actually are running a clinical trial in patients with ABS with a pain score of 4 or above out of 10. And we are running a study, and we believe that it will work quite well with patients with moderate pain. With severe pain, probably you have to combine with conventional therapies.
I'll look forward to hearing more about that study in the future. That's excellent. Thank you.
Thank you.
Do we have time, maybe, Jen, for maybe one quick answer question from perhaps Tom and Jiande? This one kind of originates from Dr. Sin out in Reno, but you know, you're both stimulating various ways, Jiande, through the auricular, vagus afferents, and Tom on the stomach, and you showed changes in slow wave. Jiande's did, I think, in particular. Do you think that signal is somehow affecting the ICCs? And how do you envision the vagus, or is it affecting some other part of the gastric wall, some other cell?
Tom, you go ahead. I think you have the data, right, with GS.
Yeah, we've got some data. Great question. Obviously, the ICC is really where things originate with the slow wave. But I think when you neuromodulate, you probably get a lot of effects, including on other cells. We have some data looking at effects on mast cells, for example.
Mast cells.
That's one thing that we have a lot of data on, as well as some other neurotransmitters. So I think it's more than just the ICC. It's the ICC obviously is connected to a lot of autonomic innervation as well, particularly in the submucosal plexus.
Okay.
So again, it's sort of like the stimulation parameters. Stimulation is kind of like a dose. It's like a drug. The energy you give is dose, and I think the effects you see probably are concomitant to that. But I think it's more than just that.
Thank you. Jen?
Yeah. In our animals models with GI dysmotility, like opioid-induced constipation, and then when we apply the auricular vagus nerve stimulation, we can somehow reversing the reduction in ICC number. So that's, and also like chatter neurons. But that is associated with disease and associated with stimulation parameters.
Did you say you actually increased the numbers of ICCs?
That's right. So in opioid-induced constipation rats,
yeah,
the number of ICCCL was reduced, but with chronic stimulation, we can increase.
Yeah, fascinating. Yeah. Well, it could be the magic of electricity. Of course, that's magical stuff, that electricity.
Yeah, because you are working on the parasympathetic and sympathetic nerves, right?
Yeah.
Excellent. Well, we've come to the end of our time for our symposium. We're out of time to answer any further questions, but any leftover questions will be answered by our presenters at Doc Matter. And we thank our speakers and all of you, of course, for attending and answering questions. Have a great evening.
Yeah, thank you. Thank you, Tom. Thank you, Jiande.